And that brings me tonight, of course, to our speaker, Professor Christopher Wills, who'll be telling us about the evolution of complexity from the human brain to the rainforest. And as ever, I find it a unique pleasure to be able to introduce scientists of the caliber of Professor Wills. He, like many great scientists, was born in the UK, and but finding that a little too small, his family moved to Canada, and he grew up there in Canada and received his first degree from the University of British Columbia, and later on received a PhD in genetics from UC Berkeley. We were very lucky to have him move to San Diego and join UCSD in 1972, initially as an associate professor, and now he is a very distinguished full professor in our section of ecology, behavior, and evolution in the Division of Biological Sciences. So you know that you just don't have to take only my word for it. Professor Wills has received many awards, including a Guggenheim Fellowship, awards from the AAAS, the Association for the Advancement of Science, for his excellence in teaching. And also he authored a book that in 1988 titled Children of Prometheus, The Accelerating Pace of Human Evolution, was chosen out of thousands and thousands of science books to be one of the 5 finalists for the 2000 Aventis Prize. The Aventis Prize is essentially the Booker Prize of science, and it gives you a taste, if you like, of the respect and high esteem in which Professor Wills is held by the scientific community. His interests in evolutionary biology are both broad and deep, and he touches on many areas in his research that help illuminate 3 of the key principles that Darwin spoke about in terms of thinking about evolutionary biology. And those 3 principles, of course, are natural selection, hereditary and the mechanisms of hereditary, and of course the fact that all populations need to have variation in them for evolution to be taking place. And so what Professor Wills will be taking us on a journey tonight will be talking about how diversity is important in populations and how at many different levels, be it genome evolution that's acting in terms of how humans are evolving, or the content of diversity in an ecosystem such as a coral reef or a rainforest, are incredibly important and part and parcel of the whole evolutionary process that results in these amazingly complex systems that we enjoy, be it the human brain or something as beautiful as the rainforest. So without further ado, I'd like to ask Professor Wills to come up and deliver his lecture. Thank you very much.
Well, thank you, Steve, for that overwhelming introduction. I'll do my best to try to live up to it. And I also want to thank the Museum of Natural History for hosting this series. It's really, I think, a wonderful venue. What I'd like to talk about tonight is one of the important aspects of Darwin's theory of evolution, which is that all populations, all evolving populations, populations that are capable of changing over time genetically, must be filled with genetic variation on which natural selection can act. Without such variation, even if the populations are variable and they look different, members look different from each other, natural selection won't work. Darwin was well aware of this and pointed out in The Origin of Species that there had to be variation on which selection could act. He didn't know the nature of that variation, of course, but he did spend many years studying organisms in great detail. One of the things perhaps people don't know very much about Darwin is that he spent 10 years of his life laboring over an enormous body of work involving barnacles. He examined barnacles from around the world, arranged them, named them, rearranged them into trees of relatedness, discovered many things about their life cycles, and also discovered, as you can see in this picture, which was commissioned by Darwin for one of his books, that this particular species of barnacle, of Balanus, comes in a variety of different types. It's all one species, but you wouldn't think so when you look at them. You might think these were all different species. Some of this variation is due to the fact that the barnacles are growing under different conditions, but some of the variation, as has been discovered since, is in fact genetic variation. And that genetic variation, as Darwin realized, is an essential part of evolution. So what I want to talk about tonight is the genetic variation that we see in our species, variation which can be discovered now, which can be understood now in great detail, in part because of the Human Genome Project. This cover that you see here is the cover of Nature from February 2001, in which the first draft of the entire human genome was presented. We still haven't got a complete genome, but they're getting closer and closer to it. And this, this draft essentially was something that for the first time allowed us to begin to plumb the true extent of the variation that we find within our species. And that variation essentially formed an important part of the cover of Nature itself. If you zoom in on the cover, you see that in fact it consists of a whole bunch of pictures of people, all kinds of people, all cleverly put together into this mosaic. So when you back away from it, you see the double helix of DNA. When you zoom in, you essentially get some glimpse, at least, of the variation that lies within our species. So tonight I'm going to be talking about variation within our species. I'm going to be talking about variation within some plant species that we'll be talking about in a moment, and also variation in ecosystems where the variation is not necessarily at the genetic level, maybe at the level of the species. We'll be talking about rainforest ecosystems, but it turns out that the processes that maintain the variation in the rainforest have a surprising amount to do with the processes that maintain variation within our own species. Just to give you a glimpse, to remind you of just how variable our species is. I've been doing a lot of traveling in connection with this research. Take a camera with me. I've taken pictures of all sorts of interesting people, and you get some glimpse at least. Here are some very determined people who live under the grimmest conditions in the central plateau of Madagascar. Many other people in Madagascar, a pantanero from southern Brazil. And this young lady is a good representative of the enormous mixture of people that one finds in that vibrant country. The Solomon Islands is a crossroads of the Pacific with many different groups of people who've come together and mixed in the islands. In Southeast Asia, a wide variety of different Aboriginal peoples, a surprising number of them, some of whom have peopled the entire Pacific, are spread over a wide range of the planet. And the variety that one sees in this part of the world in terms of behaviors, in terms of cultures, in terms of people and languages is really quite overwhelming. In India, well, everyone knows that India is an incredible repository of astounding variation. I mean, it blows you away, quite literally. And one finds in Rajasthan, in other parts of India, a huge variety of different peoples, because India, once again, has been a crossroads for many years of the migration of different peoples from Asia, from the Middle East, and Aboriginal peoples who came to India much longer ago than that. So, by glimpsing this, the glimpse, for example, of people in Australia, the Aboriginals of Northern Australia, who've lived in this part of the world for at least 40,000 years and who have had essentially a continuity of culture during all that time. And the Tiwi Islanders to the north, who once again are a mix of peoples from Australia and also some intermixture from the north. The peoples of New Guinea, well, there's a diversity for you. Huge numbers of people living in different tribal groups, more than 800 different languages spoken on the island of New Guinea, and the variety of different peoples and of different cultures is literally quite overwhelming. This child is one who will be growing up in a world that's going to become even more diverse, because now for the first time people in different parts of the world are beginning to meet each other in substantial numbers, and we're beginning to see here in America the effects of this with people like Tiger Woods and of course Barack Obama, who are wonderful examples of people who are resulting from this mixture of gene pools that had never happened or almost never happened up until very recently. Well, we can ask a number of questions about our evolution as a species. First of all, how do we differ genetically from our closest relatives? And those close relatives include the chimpanzees, who are our closest living relatives, and the Neanderthals, who are some of our closest relatives from many years ago. The Neanderthals disappeared from Western Europe, Eastern Europe, and the Middle East approximately 20,000 years ago, and it's quite possible that modern people moving into this area wiped them out. We don't know. Still, we're beginning now to glimpse some of the genetic differences between ourselves, the chimpanzees, and the Neanderthals. Second question I'd like to look at briefly. What kinds of genetic variation can be found in our gene pool? We've talked about huge amounts of variation at the visible level. Not all this variation, of course, is due to the action of genes. A good deal of it is the result of different cultures, different environments, but there is some genetic component to very many of the differences that I gave you a glimpse of in the last few lectures. What is the genetic variation? How can we characterize that variation? And third, and most importantly from the standpoint of this talk, what selective pressures have driven our evolution? And how is the genetic diversity in the human population maintained? This is a point that I'll keep circling back to as we look at some things that may not at first appear to make a great deal of sense or fit into this pattern, but it will, I think, all come together as the talk proceeds. Well, first of all, we can ask, how do we indeed differ genetically from the chimpanzees and from the Neanderthals? Well, we now have at least a draft summary of the genome of the chimpanzee. We can begin to compare gene by gene the differences between humans and chimpanzees. We find that we are about 98% identical in terms of our DNA sequences, but in terms of the genes that we have, the 20-odd thousand genes that we possess and that that chimpanzees possess, those genes are almost all held in common. You can find a corresponding gene in the Neanderthal genome to pretty much every gene that's found in the human genome, with a very few exceptions. So the resemblance then between ourselves and our closest living relatives is dramatic. Even more dramatic is the resemblance between ourselves and the Neanderthals. These people who lived in Western Europe until very recently, and also in other parts of Europe, a fair amount of DNA, much more than 1 million bases now, has been sequenced from Neanderthal bones. You can extract DNA from bones that have not become fully fossilized, and it's possible to extract this, as Svante Paabo and his colleagues have brilliantly done, and look at their sequence. This is early days yet. Paabo expects to have an actual draft of the Neanderthal genome in a year or two, and it'll be very interesting to see how that compares with us. It's clear that Neanderthals were much closer relatives to us than the chimpanzees. And the last common ancestor lived at least 500,000 years ago. So very close compared with our last common ancestor with chimpanzees, roughly 6 million years ago. So we're much closer relatives to the Neanderthals. Well, we've had a bit of a love-hate relationship with the Neanderthals over the years. Sometimes we've looked upon them as brutish creatures. This picture from about 100 years ago in a book by Marcelin Bull is essentially some notion of how some of the earliest discoverers of Neanderthal skeletons viewed these people. Brutish, hairy, carrying all sorts of nasty implements and things like that. This has lasted down to quite a while. I found this amusing little thing on the internet here. Here is a movie poster from the 1950s, The Neanderthal Man. What primitive passions, what mad desires drove him on. The guy here is really rather primitive, to say the least, and these young ladies are all running around and screaming. It is a movie. Maybe you can get it on Netflix, I don't know. But in any case, what we have discovered more recently, of course, is that Neanderthals were much closer to us in behavior and probably, as it turns out, in appearance. We now know that Neanderthals were able to, they had quite elaborate culture, their tool-using capabilities, tool-making capabilities were remarkable, they made decorative objects. One thing they didn't seem to have done was draw pictures on the walls of caves, but that could simply be because nobody had suggested it to them. So, we find that at least some Neanderthals, like many modern Europeans, carry a mutation in a gene, a melanocortin receptor gene. I won't go into the details of this gene, but it is a gene that influences the kind of melanin that is deposited in the skin and deposited in the hair. It seems likely that at least some Neanderthals had reddish hair and rather pale skin, rather like a good many Western Europeans of the present time. But the mutation in the Neanderthal melanocortin gene was different from the mutation found in modern humans. The results are the same. The origin resulted from different mutations. So the Neanderthals looked very much like us, but for a slightly different reason, and yet the same gene was involved. Very, very close. The parallels between ourselves and the Neanderthals become more and more apparent the more we learn about these people. So, what is the variation that has contributed to this evolution that has separated us from the Neanderthals and that ultimately separated us from the chimpanzees? If we look among different human individuals, we find many, many millions of genetic differences, about 80% of which are single base differences between the individuals. That is to say, if you run your eye down through a sequence of bases along a DNA molecule, every once in a while, when you compare two molecules from the modern human population, you will find, about 1 in every 1,000 bases, that there are differences between them. A single base change here that essentially is one small genetic difference. These changes, for the most part, have no particular effect, but some of them can have quite substantial effects. They can cause diseases, they can cause big differences in how people appear, and in a few cases we know about even in how they behave. So this accounts then for 80% of the variation we see. The other 20% is a huge and heterogeneous collection of other stuff. Mostly insertions and deletions, pieces of DNA that have been removed or that have been inserted into different chromosomes from the human population. So we can see many, many such differences. Our laboratories examine quite a few of these differences. Oh, I should say, by the way, that this is just a map of a region of one of our chromosomes showing in a graphical form all the differences between many different chromosomes that have been examined in a project called the HapMap project, and we'll come back to this project a little later on. What it is a survey of the single base differences in the entire human species. And the HapMap project has now tracked down about 4 million of these differences and is an astounding and extremely valuable tool for us to understand the nature of the variation in our species. We've been working for quite a few years on little repeated regions of DNA, so-called microsatellite regions, which can grow or shrink depending on mutational changes that can happen in the population. Microsatellites often fine-tune the way in which genes are expressed. And the interesting thing about microsatellite mutations is that they can happen and then they can unhappen again. It's possible for a mutation to reverse itself much more readily than a single base change mutation can. So microsatellites are just one of this other heterogeneous collection of kinds of changes that you find in the human gene pool. What selective pressures have driven our evolution? This is the third question I'd like to investigate. Directional selection is the kind of natural selection that we normally think of. Selective pressure that essentially has the effect of replacing one form of a gene with another form. One form of a gene is removed from the population by selection against the individuals carrying that gene, and another form of the gene essentially moves in to take its place, may be selected for. There's another, and this is what everyone thinks about when they think about natural selection. You're essentially replacing one gene with another. This causes a problem because people who don't have this new gene, this new advantageous gene, are at a disadvantage. They may be ill, they may be unable to have as many children as people who have the new gene. There are all sorts of reasons why there's going to be a cost to this kind of selection. As you substitute one gene for another, somebody has got to die. And this process of natural selection is one that is going on all the time. And as we'll see in the human population, it's going on at quite a dramatic rate. There's another kind of selection that people don't think of as much, and this is balancing selection. Here, new forms of a gene may enter the population and then move to what's called an internal equilibrium in the trade, some frequency or proportion in the population where they move up to that frequency and then they stop. And once they're in the population, they don't replace the old gene. They move in, remove some of the old genes are removed, and eventually you have the two forms of the gene coexisting. And you may have quite a lot of different forms of a gene coexisting in a population simultaneously. Balancing selection also has a cost, but there's a very interesting aspect to this cost. The cost is not as severe sometimes as the directional selection cost, and we'll come back to that very interesting point a little later on. What balancing selection does is pour variation into a population. The more balancing selection is going on, the more variation is being held in the population by natural selection. And that's another interesting aspect of how evolution works. I'd like to spend a good deal of time in this talk talking about a particular type of balancing selection called frequency-dependent selection. Frequency-dependent selection is selection in which a gene may be at an advantage when it's rare, but as it increases in frequency, it loses that advantage and may even become disadvantageous. Let me give you a rather vivid example of this. Actually, this is work which Josh Cohn in our group is carrying out, along with other investigators who are looking at a phenomenon called pollen incompatibility. Now, you remember from what you know about plants that the fertilization process in flowering plants involves the transfer of pollen from the anther to the stigma of the female part of another flower, and then, of course, this this is all X-rated from now on, so we won't go into the details here. But anyway, this process then is one that plants, of course, carry out all the time, and the transfer, of course, can be by wind, it can be by insects, it can be birds, bats, all kinds of things can transfer pollen from one flower to another. In plants of the flowering plants of the cabbage family and some other groups of plants, there's a very interesting process here which ensures that these plants are genetically different from these. The male plant must be genetically different from the female plant in order for the fertilization process to take place. So suppose you've got a locus, which in this case is the S locus in the cabbage family. And suppose there are three forms of alleles at this locus. We'll call them A1, A2, and A3. Now, you've got these three alleles here. The rules are like Sudoku in a way. This is a kind of genetic Sudoku. So you'll get your Sudoku brains going here and you can begin to see what's happening. The rules are simple. This has to be completely different from this. So A1 flower can mate with an A2 flower. But an A1 flower can't mate with an A1 flower. So if you have three alleles, these matings are all possible, but these matings are not. So most of the matings here are not going to work. So this population is going to suffer substantially if there aren't enough alleles at this locus. Now, I think you can see intuitively that if we introduce another allele at this locus, this changes this whole dynamic, because the first plant to have this new allele, we'll call it A4, The first plant to have this new allele is going to be at a great advantage. It's much more likely to be genetically different from the other plant and therefore mate with it successfully than plants that still have those same old 3 original alleles. And to make things fairly vivid here, I've set up a little computer simulation to show you how it works. Start with a population that has 3 alleles. This axis shows all the alleles in the population. A third of the alleles are red alleles, a third are green, a third are blue. And if you have a population where the Sudoku that I just showed on the previous slide is going on, then generation after generation, all 3 of these alleles are maintained. So I let it run for 1,000 generations or so, and then I began to introduce new alleles. And here's a new allele that's popping in here. This new allele, when it's rare, has an advantage. It rushes up in frequency until it overshoots a bit and then actually begins to go down here. But it rushes up in frequency. Now you've got 4 alleles at this particular piece of the chromosome. And now another gene appears. Another one pops in. And now it also, when it's rare, has a big advantage, increases in frequency, and eventually stabilizes. And you can add more and more alleles to this system. So, whoops, wait a minute. Let's go back. Let's try it again here. There we go. Now, I'm running the simulation here. Generation after generation, we're adding more and more alleles. You can see that each time a new allele appears, it increases in frequency and then stabilizes. We're packing the population with more and more variation. This is exactly what you see when you look at wild populations in the cabbage family. The populations are filled with these alleles, and they're held there by this frequency-dependent process. The alleles are at an advantage when they're rare because they give the plant a chance to mate with other plants. But when they start to increase in frequency, they lose that advantage. And eventually you get to the point, as I say, it's quite dizzying, where you get as many as 50 alleles in this population of 5,000 plants. That's a lot of variation that's being held in the population by this frequency-dependent process that I talked about. Now, the fascinating thing about this, and I'll come back to this at the end here, I hope everyone's still with me on this, because you're packing the population full of genetic variation, and the cost of doing so diminishes with time. To begin with, there's a huge cost. Many of the matings, when there are very few alleles in the population, many of the matings don't work. But as you add more and more alleles, more and more of the matings work. That makes sense, because the more alleles you have, the more likely it is that you'll be genetically different from the plant that you want to mate with. So the result is going to be that as time goes on, the cost of having all this variation in the population actually drops. So the burden on the population then can diminish quite dramatically, and indeed this loss of this gain of the population, that the population is now a healthier population because it is packed with this kind of genetic variation. Now this is an important point. If in fact this kind of selection is happening in the human population, this may help to explain why our population is packed with genetic variation. It's not the only explanation, but it is a very interesting one. Now, frequency dependence is found in all sorts of other places, and we got involved in this because my colleagues and I have been working for some years now on very, very complex ecosystems like the rainforest. Rainforests are, as you know, bewilderingly complex collections of animals and plants that for the most part are found in the tropics. There are some temperate rainforests, but for the most part are tropical. They're just packed with variation at the species level. If you go through a rainforest, almost every tree seems to be a different species. That's not quite true, but still you may find in a given piece of rainforest literally thousands of different species of tree. The diversity is overwhelming. Why so much diversity? What is going on? Well, it turns out that the data needed to answer this question began to be collected a few decades ago by some far-sighted ecologists, most notably Steve Hubble and Robin Foster, who, with the assistance of the Smithsonian Institution, began to look at pieces of rainforest which they established, rainforest plots that they established in various parts of the tropics. Here's the Smithsonian. We're now going to hop away from the Smithsonian, dizzyingly zooming across the planet. I borrowed this idea from Steve Kay. Dizzyingly zooming across the planet until we arrive at the first forest that was established some decades ago on Barra Colorado Island in Panama. Barra Colorado Island is an island in the middle of a large lake that makes up a good deal of the Panama Canal. It's easier to form a lake by damming something than it is to dig a big ditch, and that essentially explains why you have this big lake. Barra Colorado Island used to be part of a continuous rainforest, but now it's been fragmented because of the lake. This plot is the plot that Steve and his colleagues established back in the 70s. It's a plot that consists of a half square kilometre of pretty much untouched forest, consisting of 250,000 trees, made up of over 300 different species. We now know the names of all these species, each individual tree, and this is now being followed through 5, going on 6 different 5-year censuses. We can follow over time the dynamics of what has happened in this forest. So we're able to look at slices in time and follow the dynamics of this forest, just as we were able to look in that simulation I showed you at what happens to a cabbage population as it begins to fill up with genetic variability. What's going on in the forest? Well, it turns out that if you look at different parts of the forest, you see the results of frequency-dependent selection. Except this is now not at the level of genes, but at the level of different species. If you look in this part of the forest, and you find that a species that we'll call the red species, it's one of the 300 species of tree. In this part of the forest, this species is quite common. The darker red dots here show trees that had been recruited during the census period. So these trees now have now appeared in this part of the forest. You find that if there are lots of this red species in this part of the forest, you get relatively few recruits. If there are few of the red species in other parts of the forest, you get proportionately more recruits. So when the red species is common, there are fewer than the expected number of recruits. When the red species is rare, there are more than the expected number of recruits. And this pattern is one, it turns out, that we find in many other forests. Why should this be happening? Well, it has more to do, nothing to do with pollen incompatibility or anything like that. It has much more to do, it appears, with, there are several explanations, but one of the explanations for which there is some evidence is that when a species of tree becomes common, then its pathogens and parasites and the things that eat it begin to become common too. You know that if you've got a crowded human population, diseases are much more likely to spread within that population than if the population is sparse and spread out. Exactly the same thing seems to happen in the rainforest. So when a species is rare, it's at an advantage because its pathogens and the things that eat it also are rare. When it becomes common, it loses that advantage. And this brings you back again to the frequency dependence that we keep circling back to as we look at these phenomena. Now we can look at many different parts of the world. We can jump from place to place. Some of these forests I have visited, some of them I have not. But each one of the forests shows this pattern. This forest in Puerto Rico is a forest in the eastern part of Puerto Rico. It is quite different from the forest at BCI. It is very often hit by devastating hurricanes, and you can see the kind of destruction that happens here. Nonetheless, this forest too shows this frequency-dependent phenomenon. Then if we jump to another part of the world, racing across from Puerto Rico all the way across Africa, zooming up here to India, in southern India, the great Mudumalai National Park, part of a large complex of national parks in southern India, is once again a forest that is very different from either BCI or the forest, Luquillo Forest in Costa Rica. This forest is one that is relatively dry. It's fed by water from the nearby Nilgiri Hills. This is my good friend Datta, who is showing various aspects of this rather open, dry forest. The forest in this case is damaged by fire and by elephants. And so this forest too is undergoing dramatic changes. But again, you find the frequency-dependent process. If you then hop from India across down to Sri Lanka, or used to be called Ceylon, you come to the Thinharaja Forest Reserve. This is a forest that is part of the World Heritage Site system, and it is a much wetter forest, much denser, much wetter, as you can see here. This forest is one that is much more typical tropical rainforest. It again shows exactly the same phenomenon. Racing across from Sri Lanka, across the Indian Ocean up to Thailand. This forest is a rather different forest once again. It's open, not as open as the forest in Mudamalai, but is still, it's very remote, and it's the only forest in this entire series where tigers have survived. It's possible to take a photograph of the occasional tiger in this forest. And two more forests for us to look at. This forest in peninsular Malaysia, the Paso Forest Reserve, is one that is surrounded by agricultural land, which is beginning to encroach on this forest. Much of peninsular Malaysia, and the rest of Malaysia really, is covered with oil palm plantations, and so this forest is one that is undergoing considerable stress. Nonetheless, it's extremely diverse, with more than 800 different species of tree. As with many of the other forests in the tropics, the variety of other life that you find in these forests is quite dramatic. There are all kinds of fascinating creatures that lurk there, and if you stroll through these forests, you're just amazed at the diversity of life that you find. From peninsular Malaysia, we race across the South China Sea to Borneo, the northern part of Borneo, Sarawak, which is also part of Malaysia. The Lambir Hills National Park is, as you can see, pretty much surrounded by disturbed land, but Lambir Hills is the repository of the most diverse rainforest of the lot. Over 1,200 different species of tree in a half-square kilometre plot, and those trees, in fact it's closer to 1,400 in the latest census, those trees are enormously diverse. Again, frequency-dependent selection is found here. Sylvester Tan, my good colleague, knows everything about the Lambir plot. How he can keep about 2,000 or 3,000 different species of tree in his head at once, I don't know. And all kinds of neat insects like these lantern bugs are found in the Lambir forest. We found more recently that when small regions of the forest are followed over time, you also see frequency dependence manifested in a somewhat different way. If you look at a region of the forest and follow the survivors from one generation to the next, you find that the locally commoner trees tend to die off at a higher rate than the locally rarer ones, so that the contingent of survivors becomes, in effect, more and more diverse as it gets older and older. You essentially are making the distribution of tree species more even in the forest over time. This, again, is a process that can be attributed to frequency dependence. So we can ask, has frequency dependence played a role in our own recent evolution? If we look at the human brain, compare it with the chimpanzee brain, these cross-sections of the human brain and the chimpanzee brain show indeed very dramatic differences. There is, in both cases, you've got an extensive cortex, but the cortex is even more folded, even more deeply folded, even more extensive in the human brain than the chimpanzee. As you look at different brains from different mammals, you find that the number of layers in the cortex also increases. There's actually a difference in the number of layers, even from chimpanzees to humans. So there is a huge increase in the level of complexity in the brain of humans compared with that of other mammals. Bruce Lahn and his colleagues have shown, using a variety of different techniques, that there are a number of genes that affect or appear to affect the way the brain develops. And these genes seem to have undergone, or in fact have undergone, rapid evolution in relatively recent times. This ASPM gene is a gene, the function of which, the actual function of which we're rather unclear about, but you know if you knock this gene out, the brain doesn't develop properly. So if you destroy the function of the gene, it has a dramatic effect on the brain. What Lau and his colleagues have found is that one particular form of this gene has spread through many different human populations, and it looks as if this particular form appeared about 5,000 years ago and has spread very rapidly in the human population since that time. Other genes that show a similarly rapid spread have also been found. But it is striking that this gene and most of the others don't ever seem to get all the way. They don't go to 100%. They don't essentially take over the entire population. Is this simply because they haven't had time to do so, or is it perhaps that some kind of balancing selection is operating in the population? We don't know. Oh, it's interesting, by the way, you might ask, gee, does this gene have some effect on people? Well, they haven't found any effect so far. The different forms of the SPM gene seem to have no effect on IQ or on brain size, but it could be that since we have no idea what the gene is really doing, that some rather subtle effects are actually traceable to the function of the gene. Well, I had predicted about 10 years ago. I'm always glad that I predict things years ago. The trouble is, by the time the information, that the evidence actually appears, everyone has forgotten that I predicted it. But anyway, 10 years ago I wrote a book called Children of Prometheus, and the subtitle of the book was The Accelerating Pace of Human Evolution. I was very much at odds with other biologists on this. Everyone else pretty much had decided that human evolution must have come to a stop. Because, after all, we all have enough to eat, we've conquered diseases, we've conquered extremes of heat and cold, there's really no reason why we should go on evolving. I contended, however, that because of the increasing complexity of our intellectual environment, there's likely to be an accelerating pace in the evolution of the genes that contribute to the different ways that our brains work. And I followed this idea up, and it was, essentially this whole thing was greeted with a large yawn. But it turns out at the end of last year that John Hawkes and his colleagues came out with a paper in PNAS called Recent Acceleration of Human Adaptive Evolution. And this paper drew on that HAP map that we talked about right at the beginning of this lecture. The HAP map, you remember, was this huge repository of all the genetic variability in the gene pool of humans. It was possible, looking at the HAP map, to find about 2,000 regions of our chromosomes where apparently some gene, and in most cases we have no idea what the gene is, but some gene seems to have swept through the population, starting with a very small number of copies and increasing in the number of copies and dragging with it other genes nearby to give it this particular pattern of genes. And this particular pattern of genes is found in different in different parts of the human species, the two in fact have increased greatly in number. And this has happened over and over again. Hawkes and his colleagues claim, after doing a lot of calculations, that our recent evolution has sped up about 100-fold compared with the evolution of our close relatives, or compared with the evolution that we were undergoing before about 50,000 years ago. In the last 50,000 years, our evolution has sped up 100-fold. I'm not sure I believe that number. Other people are also quarreling with it. But even if it's only 10 or 20-fold, there certainly is a dramatic impact on the genes of our species. What can be doing it? Well, this simply shows some of the data, which we won't go into, but suffice to say that if it's possible to date some of these so-called selective sweeps, and you'll find that they really begin to get going about 40 or 50,000 years ago, and they peak fairly recently, and you don't find anything really going all the way in the population. Most of them seem to stop before they completely sweep through. It's probably because we can't detect the ones that have swept through, but it could also be because some of them at least are landing up in the population, beginning to contribute to the pool of variation in the population, perhaps from frequency dependence. Well, if we ask what is the likely process that might have happened to our species over the last few tens of thousands of years, we have to ask, what would happen if we were all the same? Okay? Here, for example, is Bill Gates, a remarkable human being, an amazing guy. He made a lot of money. He's giving it away like crazy. He will probably end up saving millions of lives in the process. Really, a fantastic human being. And we all agree that Bill Gates has some properties, some characteristics that are certainly worthy of preservation in our gene pool. Well, let's suppose for a moment that we looked at the human gene pool and asked, all right, what would happen if we fiddled with it a little bit? Oh, we all know this, of course. This is the most famous cover of an album ever, right? The Sgt. Pepper cover, in which the Beatles are presented here at apparently at the funeral of the Beatles. The Beatles appear to be about to be interred here. A large hole has been dug. And you notice they've got all these other people along with them, all these different people. Some dim reflection, at least, of human diversity. People ranging all the way from W.C. Fields to Edgar Allan Poe, all kinds of people. It's a lot of fun to search through this cover and find out who's there. But let's suppose for a moment that we didn't have this diversity. Suppose, essentially, that everybody was like Bill Gates. Now, probably the human species would go on plugging along if everybody were like Bill Gates. But the cost in such a uniform human population would be substantial. Bill Gates imagined that, you know, some primitive tribe were full of replicas of Bill Gates. Some of Bill Gates's would have to be good hunters, some of them would have to be good tool makers, some of them would have to be good gatherers, some of them would have to be good cooks. Is it possible that Bill Gates could do all these different things? Seems very unlikely. It might be that that tribe of Bill Gates's would be at a disadvantage compared with other tribes that were more diverse, and that therefore had people who could fill these different ecological niches more effectively. So what I'm really suggesting here is that while our physical environment has become more uniform, we are, most of us at least on the planet, are relatively well nourished, not all, but most of us are far freer of disease than even our recent ancestors were. Most of us are protected from environmental extremes. At the same time, however, our behavioral environment has become much more complex. Things are much more complicated. And the point I simply want to make is that because of this increase in complexity, what we are becoming is a kind of a magnet for genetic variation. As new genetic variants arise in the population, most of them are lost, most of them are harmful, but those that confer or contribute to the ability of their carriers to fit in somewhere into this complex intellectual environment, those genes will be selected for. They'll be selected for up to a point. Get to be too common, they will become disadvantageous. They'll be just like the species in the rainforest, just like those genes in the cabbage plants. Too common is bad, rare is good, and eventually you reach this middle point. And when you reach the middle point with enough of these genes, the cost to the population actually goes down. It's easier to maintain this variation in a population that's living in a diverse environment than it would be if the population were full of Bill Gateses. I don't want to pick on Bill Gates, but pick another individual and fill the whole world up with copies of that one individual. You can see that the cost to our species would be substantial. So the point I simply want to make is it's possible to look at many different complex systems, the human brain, the rainforest, all sorts of other systems that are extremely complicated, and ask, is there some common denominator here? Is there some common pattern that we can extract from all this? And I think there is such a common pattern. Now, not all the variation in the human species is maintained by frequency-dependent selection, but I have a good suspicion that a fair fraction of it is. And so, perhaps 10 years from now, the evidence will emerge that this is the case, and I certainly hope that I'm still here in order to find out. Well, thank you very much for your attention. Your question is a very sensible one, which is that while Bill Gates, has all kinds of great genes, he's only, I think, got 2 children, and so that sort of is an average sort of family size. Shouldn't he have 20 or 30 children, kind of thing? point I would simply make is this: that the person who, initiates something or invents something or whatever, or changes the world in some dramatic way, may not have a very high evolutionary fitness. Think of Isaac Newton, who had no children at all, even though he changed the world. But there are people who can benefit from these changes, okay, and can, as a consequence, in some cases, dramatically change the gene pool. Just recently, it's been demonstrated that Genghis Khan and his relatives, who benefited from inventions that they didn't make themselves, they benefited from the stirrup, which allowed them to ride horses very effectively. They benefited from the recurved bow, which allowed them to shoot. It was a very short bow, so they were very powerful, so they could shoot while they were riding. They conquered the world. We now know from fascinating genetic studies that Genghis Khan and his immediate family have about 30 million male descendants. So they, while they weren't the originators of the idea, were able to take advantage of it. And so I think, you can look at individual cases and say, gosh, here's somebody who changed the world, but his genes have disappeared. Well, maybe not. Maybe his relatives had a lot of children, or people who could take advantage of his or her inventions were then able to do much better in terms of having children. The question is, recently it's been shown that pieces of disease genes, actually pieces of bacterial chromosome, have been found in species like ourselves. And the question then becomes, is this going to add to the variation in some fashion? And the answer is yes, it certainly is. We can actually borrow from these things, and we have done in the past. One of the most amazing examples of this, of course, is the mitochondria that live in all our cells that make our lives possible. The mitochondria are found in all our cells. They are little shriveled up remnants of bacteria with little teeny weeny bacterial chromosomes. And once upon a time, the mitochondria were free-living bacteria. They came and lived in the cells of our ancestors about 2 billion years ago, and since that time, we have benefited enormously from them. So this kind of thing is going on all the time. We find it in coral reef organisms, we find it in many places. This kind of synesthesia, this kind of bringing together of, this kind of bringing together of different kinds of genes is happening all the time. Yes, the question is, I've showed a variety of different people just for fun at the beginning to remind everybody of how diverse our species is. They, as you point out, did not until very recently interact with other human groups, although there was some interaction, but there wasn't the kind of overwhelming interaction that's going on at the present time. So how then did this process that I'm talking about of frequency dependence, how did this take place? Well, it is striking that one of the things that Hawkes found in his study was that if you look at African populations and European populations, the pieces of chromosome that swept through the African population, on the whole, are different from the pieces that are swept through the European population. There was an earlier wave, which is still going on, of sweeps that happened in Africa. A later wave, as the Europeans migrated out of Africa, a later wave that is still going on in Europe that involves different genes. So in different parts of the world, different kinds of frequency dependence can be happening here. I think it's striking that so many of these genes are arising in the human species at such a rate, such an enormously rapid rate. And it's really, I think, one of the things that's emerged from these recent studies is, how can we be evolving so fast? Why have we taken off at such a huge rate? And my guess is that a lot of these genes will turn out to be the kinds of genes that I talked about, where the mutation rate can be very high, both forward and back, and these little adjustments to genes are much easier to select for because they do less damage than nasty mutations do. That's a long, complicated answer to, I think, a very penetrating question. Yes, the question is, I said synesthesia instead of synthesis. I don't know what I was thinking, but in any case, what about synesthesia? Synesthesia is this very interesting phenomenon. What evolutionary role could synesthesia have played? Synesthesia is this interesting phenomenon in which you sort of mix up your sensory inputs, and you may hear, you may see sounds as colors, all sorts of interesting kind of ways of looking at the world that can be sometimes almost pathological, but in some cases can actually lead to wonderful insights. Many artists have suffered in one way or another from synesthesia. My guess would be that the role of synesthesia in our evolution has been a highly complex one, and I suspect that genes that might contribute to synesthesia have been held in the population because perhaps they could lead to people being able to perceive the world in a new way. Consider the very important role that shamans play in so many hunter-gatherer populations. People who are able to see things that other people cannot see, hear voices that other people cannot hear. They could acquire a good deal of power perhaps in the process. And that kind of ability to transcend what we normally see is one that might have had an advantage, particularly if it was rare. If everyone was running around seeing voices, hearing voices, and nobody would think twice about it. But if they were rare, then they would have an advantage. Does that sort of... Yeah. The question is, all the different genes that we see in the human population, certainly all of them together constitute the genotypic variation of the population. To what extent is this genotypic variation related to the phenotypic variation that we see around us? The answer is the relationship is very complicated. Most of the genotypic variation doesn't matter one way or the other. Most of it is simply noise. A small fraction of it is unpleasant and is in the process of being removed from our population. Some of it is sweeping through the population and will take over other genes. Some of it is ending up in this balanced situation. All these things are going on. We know that some, but not all, of the phenotypic variation that we see can be traced to genetic causes. And the big argument has been how much of it is genetic and how much of it is simply the result of environmental differences. That argument, interestingly, is being resolved in some cases more in the direction of environment than genes, in the sense that now if you measure the contribution of genes and environment to something like IQ, it was thought that genes had a huge input just a few years ago. It now becomes apparent that genes have a much smaller input, probably no more than 30% perhaps. And that comes about because we've become rather better at analyzing the data and realizing that there are some environmental factors like the intrauterine environment, the environment before birth, that plays a large role, a surprisingly large role, in determining how the brain works. So put all these things together then, the genes are important, the environment is important. I've really been talking here about the genetic aspect of this whole thing. The environment has a huge impact as well. And further, all of us are capable of adapting, of learning new tasks, of inventing new things, of being flexible, being phenotypically plastic, as biologists would say. And that degree of plasticity is itself to some degree genetic. We have the genes that allow us to become clever in many different ways. But none of us can do everything that the species can do. None of us can be brilliant musicians and fabulous artists and superb politicians and all sorts of other things like that simultaneously. So the ability then of our species to do things greatly exceeds the ability of the individuals in the species to do things.
I'd like to show my own personal evolution by actually remembering for the first time in this lecture series to ask you to hand in your questionnaires as you leave. But far more importantly, I'd like you to join me in thanking Professor Wills for an incredible lecture.